Now we are recording. Give some reminders and some announcements. I think they're all reminders. Anyway, like I was saying on the announcement on Google Classroom, Friday is Veterans Day. So if you guys wear the red shirt, if you want to, and you do it, then you'll get extra credit. So it'll be a little bit more than it usually is. What else? Wednesday is the exam. So make sure you do it online. Make sure you turn it in by 8:50 a.m. Anything after that will be 10 points per minute that it's late. And because our exam is online on Wednesday, I figured I might as well just do lab online too. That way you guys don't have to come in at all. It wouldn't make sense to make it come in for half of a day. So lab is online too. Make sure you look at it before Wednesday. That way you can let me know if you need any help. Yeah, so that's it. I think wear red on Friday for extra points if you want, and exam on Wednesday, and lab is online on Wednesday. I think that's it for announcements. Does anybody have any questions? All right, let me update my attendance here. Give it a little bit more time for people to show up. Which is a good time. I guess I'll remind you guys again. I know I'm beating a dead horse because probably anybody who needs to hear this is not listening. But for the exams, you really should do the study guides. Like if you're doing poorly on the exams, you should probably do the study guides because I use the study guides to write the exams. And I've found generally there's a trend. Generally, people who are doing poorly on exams are also people who are doing poorly on independent work. So, it's a 2-for-1 there. Do the study guide, and it helps you prepare for the exam, and it'll also give you points for independent work. And also, as I'm sure you've seen, I posted the videos there's 2 videos of me doing the exam 3 review previous semester. So I recommend to watch those. Really, between the 2 of them, I should cover every single question on study guides 8, 9, and 10. So take a look at that. Let me know if you have any questions afterwards. And that's it. And it should be 8:01 in a second. Once it is, we'll officially get started. We'll make sure no one else shows up. Obviously, for those of you online, you're going to want to send in the attendance words sometime before 9 a.m. And it is 8:01. So let's say the first attendance word is cable, like a cable of wires, or if you have cable television. All right, let's get started. Maybe even finished with chapter 13. That would be nice. We're still on the second to last of the main bullet points, which is the evolution of populations. The last thing we talked about was the source of genetic variation. And that was basically a review because we talked about that, I think, in chapters 8, maybe chapter 9, when we talked about independent assortment, crossing over, and all that good stuff. So let's talk about analyzing gene pools. And this is what you have now. This is what we did in the last week's lab. But yeah, it's also a good time to remind you, once again, I've gone a little slow grading labs. Labs are due on Friday. I haven't really jumped into grading them yet. As long as they're there before I get around to grading them, I consider them on time. The same can be said for this week's pre-lab. But anyway, the evolutionary impact of natural selection is only apparent in the changes of populations over time. That's an important, very important distinction. Make sure you know this. In other words, another way of saying that is evolution acts on what? Evolution only acts on populations. So one single individual, again, this is very important, does not change. If it does, individuals do change, but that's not what evolution is. So, there was a lot of wrong answers on this for the lab, which makes sense. We did the lab before we did this, but people would say like, that last question about not having predators. Like, well, if there was no predator, then the moth wouldn't have to change colors. But there was no single moth that ever changed colors, right? There was no white moth that went to a dark moth. There was no dark moth that ever went to a light moth. They were all either just light or dark. And it was the population that changed over time. No individual changed. Anyway, so that brings us to the word population. I think this is the first time in the semester I'm officially telling you what it is, even though we've used the word for a while. You need to know what a population is. It is a group of individuals of the same species that live in the same area and interbreed. You'll have a slightly different definition of that when we get into later chapters, but that's basically it. The key there is they're the same species, and they're basically in the same area. Which is a little bit, you really need to think about that, because usually, if you're not a biologist and you think about the word population, usually when you hear population outside of biology, people are referring to the human population. So when you think about the human population, in a sense, we're all over the world. And technically, we all have the capability of mating with anybody on the world or on the planet. But other populations are different, right? Other species are different. So you might have a population of squirrels on WVSU's campus. You do have a population of squirrels on WVSU's campus. That's a different population than the squirrels that are in Kanawha State Forest, for example. They might be the same species, but they're far away enough apart from each other where they're not competing for the same resources and they're not interbreeding or anything like that. So are there any questions about what a population is? All right, this is kind of what I was hinting at a second ago. Different populations of the same species may be geographically isolated from each other. Like I said, the squirrels on WVSU's campus versus the squirrels Kanawha State Forest is just an example. And when that happens, the exchange of genetic material never or rarely occurs. So when you think about it, probably the squirrels from WVSU don't make it out to Kanawha State Forest or vice versa. Of course, it might happen on occasion. Who knows? Maybe a squirrel accidentally jumps in the back of the truck and it gets stuck there and then makes its way to Institute and it jumps off and here it is. But anyway, such isolation is common in populations that are confined to different lakes or islands. So this is a really good example. It's not a test question, but let's look at this slide right here. Obviously, the fish that are living in this pond and the fish that are living in this pond and this pond and this pond and this pond, those are great examples of different populations. Even if it's the same species, like, I don't know, maybe catfish. I don't know, let's say there's a certain species of catfish that lives in each one of those ponds or lakes, whatever you want to call them. They're isolated from each other. Those are different populations. Even though they're the same species, even though they have the ability to mate, like if they were together, they could mate, but since they're separated from each other, they can't mate, therefore they are different populations, right? Of course, I'm sure sometimes it does happen. If there was a flood, right? So all these fish that used to be isolated, when it floods, maybe they'd be together again. Or maybe a hawk swoops in and tries to grab tries to grab a fish from this lake, and it does, and it grabs it, and it's flying over this way, and then it accidentally drops it, and the fish lands in that lake, right? So it does happen occasionally. And you have to imagine, this is kind of going a little bit off topic from what the PowerPoint is saying, that imagine, if you will, that this whole area used to be flooded, and this used to be one gigantic lake, right? At that point, all those fish would be the same population. But then imagine what happened is the waters went down, and now we're stuck with this, right? All these separate lakes. But you can tell by looking that Lake A is a little bit different than Lake B. It's kind of similar to Lake C, which is a lot different than Lake D, so on and so forth, right? You can tell the difference. Probably some of them are deeper, some of them are shallow, some of them might have bigger predators, some of them might have more to eat or less to eat, or more oxygen or less oxygen, whatever the case is. The point here that I'm getting at is you have all these different populations that are exposed to all these different environmental conditions. So even though it's the same species, they have different environmental conditions. So natural selection is going to work on them differently. Again, maybe if there's really cold waters in A, I don't know, and really warm waters in B, then eventually there's going to be some differences in those populations, even though they're the same species. And eventually, possibly, they will then become different species. But anyway, any questions about that? All right, at the population level, biologists focus on what we call a gene pool. That's what you guys focused on the lab when we talked about the Hardy-Weinberg. And the gene pool, and you need to know this for the exam, the gene pool consists of all copies of every type of allele at every locus and all the members of the population. In other words, to me, I would focus on that last bullet point. Basically, we're talking about the entire genome of every single member of the population. So, are there any questions about what a gene pool is? Okay. I'm going to sit down. I'm just not feeling as good today. Let's talk about actually analyzing gene pools. And this is what you did in the Hardy-Weinberg. Analyzing gene pools. Your book is this example. Imagine a wildflower population that has two different varieties of blooms that are different colors, right? So we've got red ones and white ones. We've been using that example throughout the semester, red flowers and white flowers. Now imagine that the allele for red flowers is big R, and that's dominant, obviously, to the allele for white flowers, which is little r. And the genotype frequencies can be calculated from the allele frequencies if the gene pool is stable. So when we talk about Hardy-Weinberg equilibrium in the lab, that's what we're talking about here, if the gene pool is stable. And it's worth noting that technically no gene pool is probably ever stable. And we'll talk about that a little bit later. We talked about it during lab, but this is all a hypothetical situation, but it generally works. So here's the Hardy-Weinberg formula, and you do need to know this. P squared plus 2pq plus q squared equals 1. Another way of saying that is, well, I'll just show you on the next slide, and then I'll take questions. There's two different ways of looking at the Hardy-Weinberg formula. And again, basically what we're looking at is percentages. So we're talking about what we call frequencies. We call them frequencies when we use them in this context, but basically they're percentages. So if we're talking about alleles, let's say, for example, we had 75%, I don't know, big R alleles. And we know that equals 1. Then what percent is Q? I know you're the only one here. We have some people online. But does anybody know what number this would be? Right here.
25?
Yeah, 0.25, right? And that part's easy. The allele frequency is easy, right? Because it all has to equal to 1. Or another way of looking at it is 75% plus 25% equals 100. Those two equations are basically the same thing. I just wrote them mathematically differently, right? So these two things are the same thing. But yeah, that's how you can figure it out, right? If you know the frequency or the percentages of one, then you can figure out the other. But that's just for the alleles, you know, is it dominant or is it recessive? It gets a little bit trickier when it comes to genotype frequencies because now we're not just looking at alleles, we're looking at genotypes. So here we go, p squared equals the dominant homozygotes, 2pq equals heterozygotes, and q squared equals recessive homozygotes. This is what you need to know for the exam. So make sure you know this for the exam. Because this is not a math class, so I probably won't have you doing any math, but if I do, it'll probably be easy like I just did up there. But there will be a question where I might give you this equation and say 2p squared or 2pq represents what? And that represents the heterozygotes. So again, as an example, we think about those goldfish that we did in that lab last week. The dominant homozygotes were orange fish. The recessive homozygotes were white fish, and the heterozygotes were orange with white stripes, or white with orange stripes, however you want to put it. So again, that's how you can do it. You can figure out, mathematically, you can figure it out. So I'll do the same thing here. Let's say this is, I don't know, 0.25 again for the dominant homozygotes. Let's say the heterozygotes are 0.25. Then what would the recessive homozygotes be? That's 0.25, that's 0.25. We don't know this one, then it all equals 1.
0.5.
Exactly, 0.5. So that's about as hard as it would get if I give you any math. Now, obviously, we have a p squared and a q squared. So as you can imagine, it can get a little trickier. And I'm going to give you an example of a trickier one, but for the exam, I'm not going to have you doing any square roots. So it'll be nice and easy if you get any math on the exam. Just make sure you know that p squared represents the dominant homozygotes, the 2pqs, heterozygotes, and the q squared is recessive homozygotes. All right, so any questions about that? All right, I like this next section because your book points out why that's important. You're like, well, why do we care? It's not all academic. There are some reasons why we would need to know, be able to use the Hardy-Weinberg formula. And that's what we're going to talk about here. The Hardy-Weinberg formula is used to calculate the percentage of humans with alleles for certain inherited diseases. And your book gives the example of PKU, which will be the next attendance word that I'm pointing at. That is the next attendance word, if you want to call it a word or an abbreviation, but whatever. That's the next attendance word. And it's just an example. I'm not going to ask you specifics about this, but this is a great example to show you how you can use the Hardy-Weinberg formula and figure things out. Your book also tells you what it is. It's the inability to break down the amino acid phenylalanine, but you definitely don't need to know that. It's caused by a recessive allele. So that would be, if we're talking about P plus Q, which we're not, if we're doing the more complicated version, we would say that that would be the recessive allele is Q, right? We know that. Here's what we do know. We know 1 out of every 10,000 babies in the U.S. has it, right? Therefore, and again, we're looking at P squared, which is homozygous dominant, plus 2PQ, which are the heterozygotes, plus Q squared equals 1, right? That's our formula. We know this is a recessive allele, so we know to have it, you have to be homozygous recessive. Therefore, if we know that 1 out of every 10,000 babies has it, we know that Q squared, which represents the homozygous recessive, equals 1 in 10,000. You guys with me so far? All right. And when you write 1 in 10,000, what that comes out to is this. That's where this number comes from, right? So 1 in 10,000 is the same as 0.0001. Now, we know what Q squared is. So now we can figure out the rest of it. First of all, we need to solve for Q. So we take the square root. You don't see that. We would take the square root of this side. We take the square root of that side. The square root of Q squared is Q. The square root of 0.00001 is 0.01. And then from that, again, we know here, P plus Q equals 1. Therefore, this is a little bit wonky, but therefore we know Q equals 1 minus P, right? That's how you figure that out algebraically. All right, so you fill that in right here. 1 minus Q equals P. I did that wrong. Mathematically, I did that wrong, but it's still, mathematically, I did it correct, but I showed you the wrong thing. Either way, that's still correct too. 1 minus Q equals P. We already know what Q is, so we plug that in for Q. So that gives us 1 minus 0.01 equals P. Therefore, P, which is the dominant allele, equals 0.99. And then there, obviously, since we know P, then we know Q, then we can plug those things in, 2 times P times Q. And that way we know about 2% of the population are carriers. That's just an example. I'm not going to ask you anything about that. You're not going to have to recreate it. I just wanted to show you how it can be put to use, right? Just by knowing how many babies have this recessive, or how many babies are born with this recessive disease, we can then figure out how many people are carriers. We can figure out how many people are completely non-affected, all by using a little bit of algebra. So any questions about that? All right, it's a lot more complicated than what you guys did in lab, right? In lab, you just said, all right, these alleles went up or they went down. These genotypes went up or they went down. You gave a percentage to that, but then you figured out, all right, was that an increase or a decrease? All right, here we go. Again with the lab. This is stuff you already learned in lab, but a non-evolving population is known as a population that is at equilibrium or Hardy-Weinberg equilibrium. We already talked about that. And so what is the Hardy-Weinberg equilibrium? You might need to know that for the exam. The short answer is it's when nothing is evolving. But the book answer is it's when the gene pool remains constant from generation to generation, which is another way of saying it's when evolution isn't happening, right? Nothing is changing. Another word of even saying that is it's when the frequencies of alleles, which are represented by P and Q, and the genotypes, which are represented by P squared and 2PQ and Q squared, remain unchanged. So again, both of these bullet points, long-winded way of saying things don't change. Nothing's evolving. And because the generation to generation change and the allele frequency is evolution on the smallest scale, we refer to this as microevolution. So again, all those things you looked at when you did that fish lab, that was microevolution, right? We weren't changing it from one species to the next. As a matter of fact, the population wasn't even changing as far as their makeup, right? You started with orange, spotted, and white fish, and you ended with orange, spotted, and white fish, right? Now the numbers, the percentages of those changed, but that was it. So let's talk about it, even though you already learned about it in lab, the mechanisms of evolution. What mechanisms can change the gene pool? Obviously, natural selection is the big one, like I keep saying, but natural selection does not necessarily equal evolution. There are other things that cause changes. We've talked about them in lab, and let's talk about them now. The first one you need to know is this one, genetic drift, but you don't need to know them in any particular order, but it's the one we're talking about first. Genetic drift. Might as well just put, if you're taking notes, genetic drift equals chance, right? This is a change in the gene frequencies due to chance. And I used the same example on during the lab when I was talking about the orange fish versus the white fish versus the spotted fish. Let's say there was a big snowstorm, and then the predators were eating the orange fish because they stuck out. That would be natural selection, right? That's not due to chance. That's due to nature selecting against the orange fish. But if it just happens due to chance, then that's called genetic drift. And here's an example. Here's a population what do we have here? We have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 red flowers and 1 white flower. You can even see their genotype, right? That one's homozygous dominant. This one's heterozygous. This one's homozygous recessive. And we even have the numbers, right? The P's and the Q's, the allele frequencies. That's generation 1. And there's no advantage to being red. There's no advantage to being white. So it just so happens this generation, once they mate, it's the ones that are circled or squared or whatever, squared off. Those are the ones that happen to mate, not because they're better than the other ones. It's just by chance. And then they have offspring. And here's the newest generation. So now we have 1, 2, 3, 4, 5, 6, 7 red ones and 1, 2, 3 white ones. So you can see the population changed. It's going from fewer whites. Now we're getting a little bit more white ones and fewer red ones. And then of course you can quantify it and see that it's actually 50-50 now, right? We went from 70-30 to 50-50, all by chance. Again, there's no advantage to being red. There's no advantage to being white. Then just by chance for this generation number 2, the ones that procreate are that flower and that flower. And then by the 3rd generation, you can see we now have a generation entirely of red flowers. Not only are they entirely red, but by chance they are all entirely homozygous dominant, right? So again, we went from 70-30 to 50-50 to fully red. All by chance. There was no advantage to being red. That's just the way it happened to happen. And that's what genetic drift is. It's all by chance. So any questions about that? All right, here's where it gets a little bit more complicated. Genetic drift is pretty simple to understand. It's a change by chance. But where it gets a little bit more complicated is there's now 2 subdivisions. So if we're talking about genetic drift, there's also 2 other things that fall into the category of genetic drift. And those are called the bottleneck effect and the founder effect. So let's talk about them. The bottleneck effect is an example of genetic drift, as I just said, and it results from a drastic reduction of population size. So know this. Know that the population effect results from the drastic reduction in population size. That'll be the next tense word. Bottleneck. So yeah, the bottleneck effect is still genetic drift. It's all by chance. So it's a result of from drastic reduction in population size. So the example I use, just because I already started it because it's in the news, I started this on Friday. Let's say there's a nuclear war with North Korea, and we have a nuclear winter, and a lot of things die off because of the cold. Now that you might argue would be natural selection. So even though it might happen because of a drastic reduction in population size, that will probably still be natural selection because the things that survive may have survived for a reason. Maybe because they, again, I don't know, the squirrels are fuzzier, so they're able to stay warm in the nuclear winter, or whatever it may be. So not only is it, what I'm getting at here is if we're talking about the bottleneck effect, not only does it have to be a drastic reduction in population size, but it also again needs to be by chance. So it's not like nature chose the ones that died. Another good example would be, imagine if COVID was worse. And we did lose a lot of people and still losing some people to COVID. But imagine if we lost a lot of people to COVID, right? That would be a drastic reduction in population size. But again, that would not be the bottleneck effect because some people are just more predisposed to COVID than others, and that would be natural selection. Anyway, passing through a bottleneck, which again is a severe reduction in population size, does some things. First of all, some bad things. It decreases the genetic variability because some alleles are lost from the gene pool. And I'll give you a visualization for this on the next slide. But that's one of the reasons why a bottleneck is bad. And obviously, if you're losing a bunch of your population, you're going to lose individuals, right? And when you lose individuals and you lose alleles, you lose adaptability. So here's an example. Here we have this population of marbles. What we have here, purple ones, green ones, and orange ones. You can see it's, I don't know, it looks like almost about even purple and orange with just a few, about even purple and green with just a few orange. Then you have a drastic reduction in the population. In this situation, it's represented by pouring a few marbles out. And by chance, only some of those marbles are going to come out, right? There's no advantage to being purple or green or orange. This is by chance, that's what happens. Those are the ones that come out. And what you're left with is an almost entirely purple population with one green and no more oranges, right? Now, that was all by chance. And that is important because imagine if these were 3 different alleles for a gene, and let's say there's a disease that comes, and the disease really hits people or really hits organisms, whatever species this is, that have the purple allele, right? Purple allele, that's deadly for them. People with a green allele, it's slightly deadly. About half of them lives, about half of them don't live. But the people who had the orange allele maybe would have been completely immune to the disease, right? So now, because you went through this drastic bottleneck, now your new population is more susceptible to this hypothetical disease I just made up, because by chance, all the people who would have been immune to it died off in the bottleneck. Does that make sense? Any questions? All right, so knowing what a bottleneck is, that's the easy part, I think. It's in the name, right? The bottleneck. It's when you have a drastic reduction in population. Just like when you're driving on the freeway, and the lanes go from 6 lanes down to 3. That's a bottleneck, right? Because you go from a drastic change in lanes, a drastic reduction in lanes. Knowing that is easy. The hard part is understanding why that's significant when it comes to genetics. Your book gives a real life example. I'm gonna put it next to this, indicating that I'm not gonna ask any questions about this real life example, but I think there are questions on the study guide. Anyway, the real life example is that cheetahs experienced at least 2 genetic bottlenecks that we know of. The first one was in the Ice Age about 10,000 years ago, and then the other one was in the late 1800s when farmers hunted them nearly to extinction because they were trying to protect their farm animals. Anyway, so because they had 2 bottlenecks, they have very little variability, and because of that, they've reduced their capacity to adapt to environmental challenges. So again, in the example I just gave, the orange allele was the one that could have saved them from disease X. And now because the orange allele doesn't even exist anymore, then they're all very susceptible to disease X. Anyway, any questions about the bottleneck effect? All right, so remind you, bottleneck effect is still a type of genetic drift. There's 2 types that we're talking about. We talked about bottleneck effect, and next we're going to talk about this founder effect. Founder effect is still a type of genetic drift. And in a sense, founder effect is a lot like bottleneck because we're going from a large population to a small population. That part's the same. The only part that's different is this time we're not going from a large population to a small population because things have died off. In this case, it's because we have a few individuals that have colonized isolated habitats. And when you think about it, this is kind of what I was getting at when we talked about Darwin and the Galapagos Islands and how the Galapagos Islands were far enough from mainland South America where, for the most part, life is not going from mainland to those islands. But sometimes it does happen. So that's what you would call a founder effect. Anyway, this represents the genetic drift in a new colony, and it explains some relatively high frequencies of some inherited disorders in small human populations. I'm going to put an X to this because, again, you don't need to know about it for the exam. But if you download the PowerPoint and click on these, you can read about it. So the Pigalac people, I think this might even be in your textbook. Those people have a higher percentage of color blindness because by chance, the people that founded those islands out in the Pacific had a higher population of colorblind people on those boats. Therefore, the rest of the population that come after them, all their ancestors, have a higher population of color blindness. Amish, that's a whole topic. You guys should look that up for independent work. They have all kinds of things. Now, when you think about it, this is interesting because Amish people are not on the islands, right? So they're not geographically isolated like you would normally think of as a founder effect, but they are a new colony and they are isolated, right? They might not be geographically isolated, but Amish people are culturally isolated. There's not a lot of people going into the Amish communities, like not just physically, but culturally, like joining the communities. And there's not a lot of people leaving the Amish community. Therefore, they are isolated. Yeah, if you look it up for independent work, there's a lot of things that are wrong with them. One of the things I'll mention is polydactyly. So there's a higher percentage of people with extra fingers and toes in the Amish community than there are in other communities. Because by chance, the people that founded the Amish community had a slightly higher percentage of people with that. And then finally, Martha's Vineyard. This is a good example too. This is where American Sign Language was born, because by chance, the people that founded Martha's Vineyard, they had a slightly higher population of people who were deaf, and because of that, now, generations later, there's still a slightly higher population of deaf people on Martha's Vineyard than there are in other places. And of course, all this is interesting when you think about it, other than the Amish, because now when we think of humans, we all just kind of go where we want for the most part, right? People can move, and they do move, so genes are flowing throughout the planet. But they didn't used to, right? It used to be a lot harder to move around. Anyway, are there any questions about the founder effect or any genetic drift for that matter? It's all by chance. All right. Let's move forward. Talk about gene flow. This is the easiest one. There's not much to say about this. And it's in the name. Genes are flowing, right? If you can remember that, then you're good. Gene flow is another source of evolutionary change. It is the genetic exchange with another population. That's what you need to know, right? Gene flow is the genetic exchange with another population. So you should think of immigration with an I, when things are coming in, or immigration with an E, when things are leaving. Either way, it's when we're flowing between populations, genes are also flowing between the populations. This might result in a gain or a loss of alleles. I'm not necessarily going to ask you that, but that does make sense, right? Because if you're losing people or losing organisms or individuals, then you could lose some alleles. Of course, if you're having fresh alleles come in, then you're gaining them. And this could also reduce the genetic differences between populations. So imagine you had some kind of bad, there was a cruise ship filled with really tall people. Maybe the NBA did some cruise for all their people. So you have all these cruise NBA players, on average, taller than most people. They're on this cruise ship. It crashes. They get cast away on some island. And generations later, they're still not saved. So they just had this new colony of really tall people. And then somewhere else, there's the same thing happened with horse jockeys, right? So all these horse jockeys after the Kentucky Derby, they all get on a different ship. They're all on a cruise. It crashes. They're on They're on an island, isolated from each other. Generations later, they have a bunch of abnormally short people, right? So really short people on one island, really tall people on another island. And then let's say one day someone figures out how to make a boat, and they realize, hey, we're not very far from that island. So people start going back and forth. And eventually, after generations, you're not going to have an island of really tall people, really short people, because those genes are flowing. So eventually, you'll have genetic differences that are evened out between, and you probably have some regular sized people. I know that's a weird hypothetical scenario, but hopefully it's easy to visualize. Anyway, if you download this PowerPoint, you can click on that link and watch another short video about evolution. And that video includes the topic of gene flow. So again, gene flow, pretty simple, right? It's the exchange of genes through different populations, or with different populations. So any questions? All right, we're almost done. We might finish this chapter today. Now we're going back to natural selection, not because we haven't already talked about natural selection, but again, we're talking about the mechanisms of evolution. They're basically broken down into genetic drift, gene flow, natural selection, and also this should have its own main bullet point, sexual selection. We'll get there when we talk about it. So back to natural selection. This is worth noting because this natural selection compared to gene flow and genetic drift, it is only natural selection that leads to an adaptive evolution, meaning it's evolution that results in a better fit between an organism and their environment. So the rest of it, it's by chance basically, or gene flow, it's not by chance. It sort of is. So whichever organisms go from one population to the other, it's kind of by chance, you could say that. But either way, it does not lead to adaptation. Your book gives this example of the blue-footed booby. It's a bird, and it is adapted splendidly for its home. So, for example, it has a body and bill that are streamlined like a torpedo, which minimizes friction as it dives. So whether these things hunt or eat, however you want to say it, they just kind of float around, they fly around in the sky, looking at the water, looking for a fish that they want to get. And when they see one, they dive down and they get it, right? So obviously, the more streamlined they are, the better, because the quicker they're diving through the air. Also, once they hit the water, they can go in easier and go in farther, right? So that is a great example of adaptation. Also, they have a very large tail, which functions as a brake. So, if there's a blue-footed booby that happens to be born that has a really small tail, there's a high possibility it might drown, right? So it dives into the water, it tries to slow it down so it doesn't go too deep, but it can't because it has a tiny little tail, so it might drown. Therefore, that tiny little blue-footed booby dies and doesn't pass along its gene, and that small tail does not get passed down. So generation after generation, again, the tails will get larger because they function as a brake. Also, on a side note, I'll say side note here, but the females prefer the males with the brightest blue feet. Now, that's actually sexual selection and not natural selection, but your book included it, so did I. Now, if you download the PowerPoint, again, you can click that link, and there's yet another video about natural selection. This is what a blue-footed booby looks like. Again, you can see how it's streamlined, right? It's meant to be aerodynamic and hydrodynamic. You can see this big tail right here, meant to slow it down when it needs to. And of course, there's the big blue feet that the females prefer. Now, even though that is sexual selection, it is worth mentioning anyway that probably only really healthy blue-footed boobies have bright blue feet, right? So if they're sick, maybe because they have parasites, and because they're just not good at catching food, so they're starving to death, or if they have some genetic problems with them, their feet aren't going to be as bright and blue. Therefore, even though it might seem silly to think, oh, the females are attracted to bright blue feet, it's not just that the bright blue feet, for them, it's an indication of health. Even though I'm sure they're not thinking that, like, oh, those are blue feet, that one looks really healthy. It's not how it works on the mental level, but, that's how it works on the species level for the population. Anyway, any questions about that before we dive a little bit more into natural selection? Okay, let's talk about evolutionary fitness. And we've already talked about it a little bit. We've hinted at it. Now we're just going to give it some vocabulary words, basically. The first word you need to know is relative fitness. I know this word for the exam. And that is the contribution an individual makes to the gene pool relative to the contributions of the other individuals. So whenever you've heard the term survival of the fittest, that's basically what they're talking about, right? Relative fitness, if you want to use the slightly wrong term, is also known as survival of the fittest. Why do I say that's slightly inaccurate? Because it's not just survival, right? So if we're talking about the contribution to the gene pool, and there's two components to that. Survival is one. You have to survive, yes, but then what do you have to do? You have to reproduce, right? So you have all the genes in the world that help you survive, but then there's something wrong with you genetically that doesn't allow you to reproduce, then it doesn't matter, right? You still are not contributing more to the gene pool than other individuals. So again, relative fitness is contributing to the gene pool. And yes, people think of it as survival of the fittest because that is a big component of it, but the other component is the ability to reproduce. As a matter of fact, hypothetically, if some organism were born to where it could just procreate a lot, like right out of the womb or whatever, it's born and then just procreates right away. It has lots of children or has lots of offspring, but then dies right away. That would still be relative fitness, right? It would still be contributing more to the gene pool than something that lived longer, but didn't contribute to the gene pool, didn't reproduce as much. So again, it's not always survival of the fittest. That's just part of the story. Any questions about what relative fitness is? All right, here's another topic you should already be familiar with. If you did the pre-lab for the other, for the second evolution lab, then you would have watched this video where we talked about the 3 general outcomes of natural selection. And let's just skip ahead. I'll just talk about it, not do the introduction. There's 3 different types. There's directional selection, disruptive selection, and stabilizing selection. And really quick, I'm going to read you what this says, but then I'm going to quickly show you the graph because it makes a lot more sense when you see the graph. Directional selection is when the population shifts by selecting in favor of one extreme phenotype. So if humans were to get on average, I don't know, a foot taller in 100 years, or a foot shorter in 100 years. That would be directional selection, because we're really taller or shorter, right? This is the extreme of the phenotypes. Disruptive selection leads to a balance between two or more contrasting phenotypes. And that'll just make more sense when I show you the graph. And then stabilizing selection favors the intermediate phenotype. So in a sense, stabilizing is kind of the opposite of directional. Sort of. But it'll just make more sense when I show you the graphs. So there we go. Here's the original population. We have a bunch of mice. You can see some of them are really light. That's one extreme of the phenotype. Some of them are really dark. That's the other extreme of the phenotype. You can see most of them are this. Right? You can consider this. Think about this as the number of individuals, because it says frequency, but it might help you to think of it as the number of individuals. I don't know. Let's say maybe that's 1,000, right? So there's 1,000 of, that means there's 1,000 of these individuals that are medium color, right? 1,000. And then, let's see, the slightly darker ones, I don't know, let's just make up a number. Let's just say there's 500, right? So you can see there's fewer of the slightly darker ones and also fewer of the slightly lighter ones. Does that make sense? You guys understand how that graph works, I hope. Yeah, so if we're doing directional selection, or directional selection happens, that means we're moving towards one phenotype, right? So it may be darker or lighter. In this case, you look at the example of going towards the dark phenotype. So you can see there's fewer of these gray ones after however many generations, and there's more of these dark ones. There's even fewer, they're not even showing them. There's like no more of the light colored ones. They're gone now. That's directional selection, right? We move from one section to the other. To give some sort of example, imagine this population of mice lives on a bunch of rocks, and there's light rocks and there's dark rocks. So the ones that are medium colored right here, they're the ones that survive the most because they kind of blend in, because light and dark equal gray, right? And then let's say, I don't know, there's a big volcano, so everything's covered in dark rock after that. Because of that, these light colored ones are going to stick out to the predators. They're going to be eaten more. The darker ones are going to survive more. Therefore, the population is going to shift. And you're going to have more darker ones, fewer lighter ones. But any questions about directional selection? All right, for the exam, you're going to get some random situation where I say, this is what the original population was, and they changed to this. Was it directional, was it disruptive, or was it stabilizing? So as you're studying this, understand that's how the question's going to go. I'm going to give you a hypothetical situation. You're going to need to tell me which one was which. Also, before we move on to disruptive selection, I should point out this that we're describing here is basically what happened with the moths, right? We went from lighter colored moths to darker colored moths. That was directional selection. All right, disruptive selection. In this case, we're getting an increase in the light colored ones. We're getting an increase in the darker colored ones. Only decrease we're getting is the one that was in the middle, the one that used to be the majority, which is the mid colored ones. I wish I had more time to talk about an example of that. 841, I can probably pull it off. All right, so there's a certain beetle that's called a sneaky bastard. That's not what the beetle's called, but the name of the situation, I forget, somebody called it the sneaky bastard effect. And what happens is these beetles mate. They fight for their mates. So the bigger the male is, the more likely it is that it's going to win the fight and be able to mate. So that selects for larger beetles, right? So that would be, that's directional selection because you're going larger. However, other beetles are really, really small. And the smaller they are, the more they look like females. And what happens is while the big ones are fighting, the small ones go in and mate with the females. So these two big males are fighting over the female. And this other one, the sneaky bastard, as they say, sneaks in and mates with the female while the other two are fighting. So that in itself, by itself, you would say, that's directional selection because that's selecting for smaller ones. Yes. So if either one of those things were happening separately, that would be directional selection. But since they're both happening, since we are now selecting for the extreme of the phenotypes, in the example I just gave, bigger or smaller, and in this picture, lighter or darker, that is disruptive. So any questions about disruptive selection? All right, and then finally, the other example is stabilizing selection. Now that I'm looking at the graph, a better thing to say is stabilizing selection is essentially the opposite of disruptive selection. So in disruptive selection, you're having more of the extreme phenotypes and fewer of the middle phenotype. disruptive selection, you have more of the middle phenotype and fewer of the extreme phenotypes. And if you watch that video that was in the pre-lab the other week, a good example of that is human birth weight, right? Because a baby that's born too small is not very healthy and is less likely to survive, so you don't get very many small babies. Likewise, a baby that's too large is going to have complications in the birthing process, and therefore, you don't want to be too large. So there's a sweet spot for baby sizes, and that's worth looking into for independent work. Because I know they mentioned it in the video, but you can look it up and get some more specific information. Just think about how big the differences between human sizes, like even cultures or populations of humans. Some people are really short, some people are really tall, yet birth weight around the world is generally pretty narrow. Anyway, any questions about the different types of selection? Again, for the exam, you're going to need to know, I'm going to give you a hypothetical situation. You need to recognize it as directional, disruptive, or stabilizing. All right, if there's no questions, we'll move on to the last bullet point of this chapter, which is sexual selection. We've already kind of gotten into it a little bit, but let's get into it some more. Sexual selection is when individuals with certain traits are more likely than others to obtain mates. That's the book definition. But think about it, it's a little bit more than that. It's not just that, because it's a matter of, it's more like they have the certain traits, but also those traits don't really help in survival. So again, those blue feet on the blue-footed booby, that doesn't help those things live. It might be an indication that they're living well and they're healthy, but the blue foot itself does not help those things live. Same thing, well, I get some more examples as we move forward. Make sure you know what sexual selection is. That brings us to another term, which is sexual dimorphism. And that is when there's a distinction in appearance between males and females, right? A great example of that is birds. Generally speaking, the male birds are all bright colored and, really fluffy, and the females are usually a little bit dull, because it's the females that are selecting the males, and the males are the ones that need to put on that display. And again, like I said here, so when we talk about sexual dimorphism and sexual selection, it's not directly associated with reproduction or survival. So again, we're not talking about survival of the fittest or contributing to the gene pool necessarily, not directly. Because all these things that we look for in sexual selection, again, they're not directly associated with survival. If you download the PowerPoint, click on this, you can watch yet another video about evolution if you want. But here's another great example of sexual selection. The peacock, and I mentioned this earlier, that's what that big tail is for, right? The females look for that big tail. And that is only to get a mate, right? There's no other advantage of having that big tail. If anything, it's actually disadvantageous. It's harder for them to hide from predators, and it's obviously harder for them to get away from predators, right? So it's a great example. Anyway, your book points out that some species have what we call secondary sex structures that are used to compete with members of the same sex. So the peacock is not an example of that. A good example of that would be, and I'll come back to this slide, but this right here, right? So these guys are competing, right? Anyway, so that's what secondary sex structures are. And then your book also points out more commonly individuals of one sex, and it's usually the females, are choosy and selecting for mates. And that's why we have sexual selection. And it makes sense. And it makes sense that the females are more picky, because theoretically, males can just give sperm all day, every day, their whole life, well, once they're making sperm. Females, on the other hand, right, they have limited number of eggs. And very often, depending on what kind of animal it is or organism, but very often it's the female that's taking care of the offspring. So it makes sense that the female is the choosier when it comes to choosing mates. Anyway, any questions about sexual selection? Here's some more examples. We already pointed out these mates, these males that are fighting for a mate. And here's a great example of sexual dimorphism with these birds. There's the one on the left here is a female. You can see this more brightly colored one is a male. So there's sexual dimorphism. Same thing, and I wish I had more time to get into it, but even plants have sexual dimorphism. You can see like a lot of plants are both male and female, and you can look and see what the male part is and the female part, like pine cones. You can tell the difference between the male and the female pine cone, which you can look up for independent work if you want. The next word for attendance, circle it, put an arrow there, and yet another arrow. So that is actually the last word for attendance. because the next thing we're talking about is going to go very quickly. Every chapter ends with the evolution connection portion, and we always skip it. Even this chapter that is about evolution. But I would say you should read it anyway. It is important. Not all the different timeline things, but I kind of talked about this earlier earlier last week when I said when you take antibiotics and they tell you to take the whole thing for like 10 days or whatever, you should do that because what happens is we're using antibiotics a lot, and what we're doing is we're causing natural selection to happen. We're getting rid of all the weak bacteria. So now we're getting these superbugs that are immune to our medicines, and that's really, really dangerous. Anyway, that is the end of this chapter. So, any questions? Great. All right. I will be online for office hours